Ecological communities are, by definition, multi-species assemblages. So, analysis of ecological communities involves data on multiple species from multiple samples or sites, and often with accompanying environmental data. And many of these variables are correlated with each other. For example, suppose you went to 10 sites and you tallied species presence and abundance, and you found 47 species overall, but with differences at different sites. You also measured environmental variables at each site, and you figure that perhaps species respond to one or more of those variables. So if you're just interested in one variable at a time, you can make something like a box plot, like we've done several times now. If you want to know how species respond to two variables, then you can do a scatter plot. But for three variables, you'd need to make a 3D plot, and beyond that, you wouldn't have the physical space to actually represent your multivariate data. Determining which factors account for the patterns of species abundance and distribution thus necessitates an approach that allows us to summarize multivariate data into fewer dimensions or axes than your original data set, and then graphically display these dimensions to detect patterns in your data. There are many different ways of doing this, with different approaches that are appropriate for different objectives or for different forms of data. They are all collectively called types of ordination. Ordination orders your data into fewer dimensions to allow you to see patterns in a form that you can visually comprehend. It is a crucial tool in the analysis of ecological communities, and we're going to cover 4 of the most widely used forms in the coming weeks. So, ordination is a family of techniques that are used for simplifying complex multivariate data sets. It's based on the principle that much of the variability in a data set is concentrated on relatively few dimensions or axes that exist because most variables are redundant to each other. And so the ordination extracts dominant underlying gradients of variation that consist of combinations of interrelated variables. And what it's doing is it's organizing your sampling entities, whether they're species or sites or observations or all of the above, along those gradients. In so doing, it condenses a large number of your original variables into a smaller set of synthetic or composite variables with a minimal loss of information. The focus is on identifying what are the dominant drivers and focusing on them. So, for example, you start out with a data set that is n entities times p variables. You can think of this like, for example, a site by species matrix. And what you wind up with in the end is an n by k matrix, where k is the number of dimensions that is comprised, these dimensions are comprised of your data, of your variables, of the most important ones that exist in such a way that your data can be ordered along them. This is very difficult for us sometimes to picture in our minds because we're so used to thinking about things in 2 dimensions, like a 2-dimensional plot. And yet this really exists in a multi-dimensional space. And we are going to display the ordinations in 2-dimensional plots. But we need to think about these things in a different way. So I'll be showing axes of variation, and you'll have to sort of detect how your data fall along those axes. But it's not the same thing as just directly plotting your data on the axes. You are synthesizing variables together to form dimensions, and the observations fall along those gradients, those axes. There are many, many ways of doing so, and so it's important for you to understand which methods are appropriate given your data and given your research objectives. There are two main groupings or divisions of types of ordination, which I show here on these two sides of this table. Unconstrained ordination and constrained ordination. Unconstrained forms of ordination are conducted on a single set of interdependent variables with the purpose of extracting the major patterns among those variables, irrespective to any relationship that they may have to variables outside of that single data set that you're focusing on. So this type of ordination, the sampling entities are not constrained by any other variables. So that's why it's called unconstrained ordination. In contrast, constrained forms of ordination seek to relate patterns in one data set to variables in another data set that may drive patterns in the first data set. In other words, variables that may constrain patterns in another data set. And so that's why they're called constrained forms of ordination. Unconstrained ordination is sometimes called indirect gradient analysis because you are looking indirectly at what causes the patterns in your data set, whereas constrained ordination, you are directly looking at drivers that could potentially account for your data. Sometimes people think about this as if you only have site-by-species data, you do unconstrained ordination to try to winnow down a huge number of variables into the most important set. And yes, it can be used for that. But that doesn't mean that if you also have environmental variables, a site-by-environment matrix, that doesn't mean that you can't do unconstrained ordination on them. And we will go over that pretty soon, and we'll have demos of that in upcoming lessons. So the difference is whether or not you're really dealing with a set of dependent variables, a set of independent variables. That interrelationship is the focus of unconstrained ordination. When you have what you think is a relationship that is also affected by some environmental gradient, then that is a constrained form of ordination that's needed. And so there are many, many different types. These are just some of the most common ones here. And so principal components analysis and non-metric multidimensional scaling are ones we're going to be doing in upcoming weeks, types of unconstrained ordination. But there's also reciprocal averaging and detrended correspondence analysis and principal coordinates analysis and so forth on the unconstrained side. And then on the constrained side, we're going to do redundancy analysis and canonical correspondence analysis. But there's also other types as well. And the choice of which ones you should use, I've got some information in the online notes about the pros and cons of each one of these methods. But in the next slide, I want to discuss how you need to go through this thought process in choosing which type of ordination to use. In order to select an appropriate ordination method, you have to consider three things. First, the type of data that you have. So, for example, if you have presence-absence data, then there are going to be certain types of analysis that are going to be closed to you. The distance matrix that you have or that you want or that you can use, because certain types of ordination are constrained by the distance matrix that you can use. And I'll talk about distance matrices in an upcoming slide. And then finally, what your objectives are. Are you really looking at for an unconstrained kind of relationship, or do you think there's a constrained relationship that's going on? So in the online notes, I had a reference to a paper. This paper right here. In fact, that is in molecular ecology, and it's about microbial communities. But it's really kind of a review paper that sort of says, okay, hey, look, y'all, in microbial communities, we've got a buku of data. We've got a lot of species, and a lot of sites, and we've got a lot of potential environmental variables. And depending on what your objectives are, there are all these different ordination methods that you can use. And so which one you should use really is important to know. And so this paper does a really nice job of going through the types of data that are needed for different types of ordination. So I highly recommend this paper. I also include some descriptions in the online notes, however, including some what I call if-then statements. If you have this, then you need that, or then this type of ordination method is appropriate, or this one isn't, and so forth. And then finally, I include a dichotomous key that was developed by Michael Palmer at Oklahoma State University, which I will go through in the next slide. This is that dichotomous key, which I also include in the online notes. And by no means is it comprehensive. It just hits the main forms of ordination. And so, for example, things like redundancy analysis and detrended canonical correspondence analysis, canonical correspondence analysis, principal coordinates, NMDS, and so on and so forth. And so this is just a small grouping of the different types of ordination methods that there are out there. In general, PCA, NMDS, canonical correspondence analysis, CCA, are probably the most common forms of ordination that are used in ecology. PCA is also probably the most misused form of ordination in ecology, which we'll cover in an upcoming lesson on PCA. But anyway, these are the main ones, but you should look over the descriptions of the other types in the online notes. They may be better suited for your objectives. Ordination is predicated on similarity or dissimilarity among variables. That's how it orders things. Things that are similar versus things that are dissimilar. And so distance in quotes here, is a concept of the proximity of objects in a high dimensional space that's defined by measurements of the attributes. This is not the same thing as how the word distance is normally used to indicate spatial proximity. In practice, the terms similarity, dissimilarity, and distance are sometimes used interchangeably, but they are not, in fact, the same. And in the online notes, I mentioned some distinctions. However, I tend to use the term distance as a blanket term covering distance, dissimilarity, and thus, by extension, similarity, as do most people in ecology. We talk about typically distance metrics or dissimilarity metrics. And in the lesson for today, we have three different packages. We have base R actually in two packages, vegan and lab DSV, and different ways of calculating distance metrics or making distance matrices from your data. And there are all of these different kinds over here, and there are many others that I haven't had time to go through. These are the major ones here. Euclidean and Bray-Curtis are probably the most commonly used. Bray-Curtis is probably the most commonly used of these. And each of these has its own properties and so forth. And you can see that certain packages only do certain ones. And so if you have need of, for example, a chi-square matrix, which handles binary data very well, then you need to go to this package, lab DSV, for example, in order to calculate it. So I spent some time in the online notes talking also about what distance metric you should choose. And what you should do is you should get into the practice of reviewing your data and then going through this sort of exclusionary list, as I did in the online notes, and we'll do in upcoming lessons as well, in order to be able to say, okay, because my data look like this, that means that this metric is not so good. So for example, if you have lots of zeros in your data set, true zeros, I'm not talking about missing data, real zeros, if that's more than 20% or 1 out of 5 observations, then the Euclidean metric is not so good, does not hold up very well. So things like that I talk about in the online notes to help you choose which distance metric that you're going to need. And so there's two big choices you've got to do. You've got to choose the method, the ordination method, and you've got to choose the distance metric as well. So in choosing which distance metric you should use, I've listed here some of the most common ones, and I've color-coded them. The green ones are ones that are most commonly used, then these yellowish ones are used somewhat, and then others that are seldom used I don't even bother talking about here. And you can see the traits of each of these metrics. There are several metrics that may be appropriate for your data set and will yield equally valid results, but they will yield slightly different ordination plots. And plots is one of the end goals that you're after. So what I want to do is I want to go through now examples of each of the 4 forms of ordination that we will cover in the coming weeks. These are just examples, just to show you the kinds of ordination plots that you'll be seeing a lot of, and then how you interpret them. So first up is PCA, or Principal Components Analysis, which is an unconstrained form of ordination. And I did this on the bryceveg.r file, the vegetation site by species matrix for the plants from Bryce Canyon National Park. And here is one of the plots. These types of ordination plots are your end goal because they allow you to visualize patterns in your data that you would otherwise not be able to detect. These analyses don't spit out a p-value, although there are analyses that we will do that will indicate whether a plot is a valid representation of your data or not. Instead, in ordination, what you're doing is you're having to look critically at what the output may indicate. There is some danger of confirmation bias when you do so, so try to be very, very neutral if you can in evaluating your resulting plots. So here in a PCA plot, the first 2 axes here, PCA 1 and 2, collectively explain most of the variation in your data. And each one of these axes is a combination of site and species data. In this graph, it's only showing the sites. These circles, each one of these circles is one of the 160 sites in the Bryce Canyon vegetation data set. And you can see, pay attention to just PCA 1 first. You can see that there is some horizontal separation along this first axis. And now pay only attention to PCA 2. There is some separation on this vertical axis, but less than this horizontal separation. And that kind of makes sense because in PCA, the axes are automatically ranked. PCA 1 accounts for most of the variation in your data, then PCA 2, then 3, then 4, and so on and so forth. So it's not surprising that when we plot the sites on in this ordination that we get more separation here than we do here. So these are just the sites. when we plot the species on this, we get what's called a biplot because it shows two sets of variables, sites and species in our case. In this graph, the sites are no longer the circles. They're black text, the site names here. And the species are in pink with these pink arrows. The site names occupy the same physical place on the graph as in the previous graph. This graph is kind of a mess because remember there were 160 sites and there were 169 species. But you can see based on arrow placement and length that some species are more closely associated with certain sites than with others. And so there's a sort of community grouping that falls out. Detecting a pattern like this in your data should then spur questions about it, like why? Why does this pattern exist? Is there some kind of environmental factor at those sites that favor certain species while excluding others? So in Bryce Canyon, the sites varied in elevation, in slope, in aspect, in soil depth, and in other environmental factors. To examine how these environmental factors could possibly explain this pattern, these patterns here, then you need to do a constrained ordination. Like RDA, redundancy analysis. A constrained ordination is needed if you want to examine the influence of environmental variables, like from the Bryce site.r dataset on your site by species data. In this case, in this example, I only examined one such environmental variable, elevation, and I just picked that. So here's the plot. And in this plot, the first thing you might notice is that, once again, we have black dots, and those are the sites. The circles are the sites. The red crosses are the species. And the blue arrow is the environmental variable that we are examining, elevation. The axes here, notice it's RDA and PCA. This is one form of redundancy analysis, and in an upcoming lesson we'll do multiple forms. But this is a great example of how you can take an unconstrained ordination result, PCA. You took your big data set and you winnowed it down to these two axes that explain most of the variation in your data. And then you use them as the dependent variables in a constrained ordination, like RDA. And the independent variables were something like elevation, like in this case, to see how much elevation affected your original ordination. So this is a great example of how you can use an unconstrained ordination and you can still examine environmental variables with it. Notice up here there is a species that looks to be an outlier up here. There is a positive association between the constrained axis, the RDA1, and the first residual axis, PCA1. But it's not strongly associated with elevation, indicating, this versus this here, indicating that there are variables other than elevation that account for patterns in your community. And again, this was just done for an example to teach you how to start reading these graphs. RDA was a constrained form of ordination. NMDS is an unconstrained form of ordination, kind of like PCA. But even unconstrained ordinations can be used to provide a quantitative description of the major ecological gradients of variation among sampling units and can portray sampling entities along continuous gradients of maximum sample variation. And it doesn't assume a linear response, the way that PCA and RDA do. And that's one of the strengths of NMDS. So I don't want you to think that you can't use unconstrained forms of ordination to examine the effects of specific environmental variables on ecological communities. You can, but the results are going to be correlative and cannot be used to indicate cause and effect. A constrained ordination is preferable when you want to look at cause and effect. But here's another example of this where you're going to have axes that can be used as dependent variables, and the axes themselves, not the original data, and then the environmental variables are going to be the independent variables to see how much these influence your ordering of your sites and your species here. So in this example, I used it to examine the effect of soil depth on community responses. I don't anticipate soil depth to be a linear variable, and that's okay with NMDS. But because depth is a categorical variable, it's not on a continuous gradient, and so NMDS might not have been a really good choice for examining soil depth. But for demo, let's roll with it, okay? So for this, I used the Bray-Curtis metric as my distance metrics. And the circles are sites that are separated on the basis of species composition. The ellipses are confidence ellipses around each of the two soil depths, shallow and deep. You can see that there is considerable overlap in the ellipses, which indicates that soil depth is not a factor that accounts for differences in plants by sites, or that NMDS was not an appropriate choice for this type of analysis for these data. Constrained forms of ordination, like CCA, canonical correspondence analysis, are superior in trying to detect the effects of environmental variables on your site-by-species data. And CCA is one of the most widely used forms. It's not perfect. It assumes, for example, a monotonic response of species to environmental variables. And by that I mean a monotonic response is it's ever increasing or ever decreasing. It doesn't necessarily have to be linear, but it's not something that's like a unimodal response along a gradient. And that kind of assumption may not be realistic for certain variables, for certain gradients. If you have drastically different sites, for example, that differ drastically in their environment, then you may well be violating that assumption. But in general, CCA is quite robust, and therefore it's very commonly used. In this example, I used it to examine how elevation and slope structure plant species composition in Bryce Canyon. The result is a triplot. It has three pieces of information. The species are the red crosses, the circles are the sites, and the blue arrows are the environmental variables, just like we've seen already. The angles between the arrows represent associations or correlations between environmental variables. So in this case, you've got better than a 90-degree angle between slope and elevation, indicating there is no strong correlation between elevation and slope, and we wouldn't expect there to be one. The direction of an arrow indicates the direction of maximum change in each variable. And arrow length indicates the importance of each variable. The positions of the circles and the crosses relative to the arrows indicates the environmental conditions at each site, and the locations of these species points relative to the arrows indicates characteristics of the ecological optima for each species. In this case, in this example, it looks like axis 1 is associated most with increasing elevation, and axis 2 is associated with decreasing slope. There appears to be more separation of sites by slope than by elevation because there's more vertical separation than horizontal separation in this triplot. So for your assignment for today, what you're going to do is you're going to take the ground beetle data, the ground beetle abundance data, and the associated simplified site information, site by environment information from GB site. And what you're going to do is you're going to go through the logical process of determining what distance matrix is going to be appropriate, or what distance metric, and then use that to make a dissimilarity matrix, and then perform PCA, RDA, NMDS, and CCA just to get some plots, like we did like I explained in the previous slides, and as I went through in the lesson for today. And think about what each of them is saying. Are their results consistent? And if not, how do they differ? And then you also can think about things like which was most appropriate, which would be the most appropriate given your data.